Hi, my name is Natalie Breska, and I'll be presenting our group lab project on the gut microbiome. So, a microbiome is a type and proportion of microorganisms living in a given location. The gut microbiome contains tens of trillions of microorganisms with more than 1,000 different known species. Although the presence of more than 50 bacterial phyla has been detected in the human gut to date, the microbiota are dominated by only two, which are the Bacteroidetes and the Firmicutes. There has been research that has determined there to be a possible correlation between immunity and the gut microbiome in a study that compared the gut microbiomes of middle-aged and elderly individuals. The finding of the research concluded a negative correlation between the presence of allostipes and age in both groups, a negative correlation between the middle-aged group and the presence of Disovibrio and Faecalibacterium. These findings show that aging could correlate with the composition of the gut microbiota. Other research has identified a relationship between the gut microbiome and brain function in a study where the composition of the gut microbiome was different in patients with depression. A negative correlation between the presence of Faecalibacterium and the severity of depression symptoms was found. This study concluded that modifying the gut microbiome can provide benefits for preventing and treating brain disorders. Acknowledging that the gut microbiome varies among the same species, our hypothesis was that there would be a significant difference in the gut microbiome of dogs and horses. So we compared 2 horses and 2 dogs. All of them were neutered males in very good health, and the horses were thoroughbreds. They had extensive exercise and only lived around horses outdoors. And the dogs were golden retrievers, and they had regular exercise. So the first step in our methods was to use the QIAGEN kit to isolate the DNA from the fecal. The isolation is based on homogenizing the stool sample in a buffer that prevents enzymes from degrading the DNA, binding the DNA to a filter embedded in a small column, and using a series of rinses to wash away impurities, and finally unbinding the DNA from the filter in a special buffer that weakens the bonds between the DNA and the filter. We then use the Zymo kit to concentrate the DNA. The DNA purified from the final steps with QIAGEN kit are again bound to a filter in a Zymo column, rinsed of impurities, and then eluted in a special buffer, but at 1/10 of the volume of the initial purification steps. So the DNA is 10 times more concentrated than it was initially. So we then use a nanodrop spectrophotometer to tell us the purity of our samples. After that, we amplified regions of the 16S ribosomal RNA gene using the polymerase chain reaction. Specifically, we amplified the V3 and V4 regions because they are species specific. PCR is conducted in a thermocycler, which is programmed to change the reaction temperature quickly. There is the first hold, 25 cycles, the second hold, and the third hold. Then we did electrophoresis, which is a technique that relies on the ability of charged particles in a solution to migrate in an electric field. The distance a DNA molecule migrates is inversely proportional to the logarithm of its molecular weight. So knowing this relationship allows us to determine the size of the DNA fragments that we are studying. We then use the agent core cleanup kit to clean up the amplicon from our amplification PCR reaction. The cleanup removes the components of the amplification PCR buffer as well as the unused forward and reverse primers. If they were to remain in solution with the amplicon, they would interfere with the indexing PCR by binding to the indexing PCR primers. Next, we did the indexing PCR step. Our PCR reaction only ran for 8 cycles as opposed to the 25 cycles used in the amplification PCR. So this process was used to add a unique set of short sequence tags to the ends of amplicons from each sample using an indexing PCR reaction. So when the DNA was sequenced, we were able to know which sequence came from which samples through those tags. When we did a second round of PCR cleanup, that just made sure that it was all clean and pure. So after that, we use a fluorometric assay to determine the concentration of our amplified sample after the PCR cleanup. Fluorometric assays are more accurate than spectrophotomic assays, and we require a more precise measurement to prepare the proper dilutions for the sequencing reaction. Fluorometric assays are based on the shift in fluorescence particles of molecule after binding to the target. So after we did this, the nucleotide sequence of each indexing PCR fragment was determined using an Illumina MISC Next Generation DNA Analyzer. So these are the results of our gel electrophoresis. So we could see a significant line in the positive controls and two of our samples, and then the rest of them were, they didn't have enough DNA for us to continue with them. So here we can see the number of successful reads for each of our samples. So for a horse sample, we had 38,781. The second horse sample, 9,730. The first golden retriever, 9,768, and the second golden retriever, 2,961. Then we have our concentrations based on the nanodrop spectrophotometer, and then we have the differences in the kingdom, order, and genus of the reads for each of the samples. Here's our results in three graphs. So here we see the differences in the genus, the order, and the kingdom between all of our samples. The other and unclassified sections are bacteria that couldn't fit in one of these phylums. So the DNA extraction from the scat was successful for two of our four samples, as the concentration was between the range of 1.5 and 2.2 at an absorbance of 260 over 280 for every sample. The gel electrophoresis indicated that only two of our samples had enough DNA after amplicon PCR, which were our sample 332 and 885. The positive control along with samples 332 and 885 were 550 base pairs. The negative control and the other two samples, 442 and 430, did not have any product bands, which show that there was little to no DNA fragments in those amplicons. The number of reads to classify the sample as successful is 1,000 or above, and all of our samples that we had originally did not have enough reads to be considered successful. So because of that, we used four samples from previous years that had met or exceeded the minimum number of reads. So for the horse sample, it had 38,781 reads, which is an adequate number. The horse sample 408 had 9,730 reads, and dog sample 386 had 9,768, and dog sample 427 had 2,961, and all of these reads were above the minimum reads of 1,000. The genus levels, when we're classified between the two samples, had the most differences. Besides unclassified at genus levels, samples 331 had the most reads of Blatia, Treponema, and Provotella, while sample 408 had the most reads as Oscillospira, Bacteroides, Candidatus, Methylacidiphyllum. The most diversity was found at the genus levels of the two dog samples. Sample 408 had the highest percentage of total reads of Bacteroides and Prevotella, while sample 386 had the highest percentage of Cadum bacterium and Escherichia. Between the two species at the genus level, there was a lot of diversity of the bacteria. The horse samples had the most reads of Clostridioides and Bacteroides at the order level, while the dogs had the most reads of Aerocypitricalis, as well as the other two bacteria previously mentioned. So in conclusion, all four samples had completely different bacteria at the genus level. The question of whether there would be bacterial diversity between the two species we chose has been answered through this project. There's not only diversity between the two species, but between the two different samples from the same species at the genus level only. So that is it, and here are my sources. Okay, thank you very much.